This video is for the Biology for Health Majors class. We're in week 1, unit 1. We'll be talking about biology, which is the study of life, the process of science, and evolution. In this particular video, we'll be talking about the process of science or the scientific method. All right, so to introduce science, science is not a set of facts. We usually think of it as a set of scientific facts, but it's not a set of facts. It's a method for exploring the natural world. We can learn facts using the scientific method, but the facts may change. They might be adjusted in light of new observations made through the scientific process. And change is good, right? Adjustments are good, because if we never adjusted our understanding in light of new data, we'd still be back in the cave ages, believing lightning was magic created by some god without an understanding of how to harness electricity for electronics or to power a light bulb. I mean, we'd have so many outdated beliefs if we never adapted in light of new information. So there's two different types of truth that, and this is sort of like a little mini lesson in philosophy, but there's these two different styles of truth that are important to understand in understanding the scientific method. There's literal truth, and this is what we normally think of as truth. This is the completely accurate, real way that the natural world works, unflawed truth. And then there's functional truth. The functional truth is our working knowledge, our current understanding of the truth that we use to operate on a day-to-day basis. For example, so first of all, functional truths can be convenient. Why would we believe in a functional truth? Sometimes we might operate under a functional truth because it's convenient, such as the example of using Newtonian physics in many engineering projects. Newtonian physics, we now know, is imperfect. The equations that arise out of Newtonian physics are not perfect descriptions of how the universe works. Newtonian physics has been replaced by Einstein's theory of relativity. But Newtonian physics are still used by many engineers for certain kinds of projects. And why? Because Newtonian physics equations are very simple compared to the equations that arise out of the theory of relativity. And they function perfectly well in conditions found on Earth. Maybe they don't function that well to describe things we observe in the universe in other parts of the universe, but they work perfectly well here on Earth. So it's a functional truth. It functions, it's convenient, but it's not a literal truth. Sometimes we operate under a functional truth because of emotional biases. A functional truth might be comfortable or familiar, and we may be attached to it as a version of our truth. We also operate using functional truths because our understanding is limited by our own technological and biological limitations, which prevent us from accessing literal truth. So what are some limitations that prevent us from accessing literal or absolute truth? Well, the most obvious would be our biology. Our biological organs are very limited. For example, here's a graph of how much of the universe we can observe through sight and sound. So on this axis here, on it's the vertical axis, the y-axis, this is the electromagnetic spectrum. That's the spectrum of light. And this narrow band between these dotted lines, this is visible light, light that we as humans can see. We can see so little of the light that's out there, so there's a lot of the universe and even our own planet that we can't detect with our eyes. There's a lot of light on this planet that our eyes can't detect. And then here on this axis, we have sound frequency. So here represents frequency of sound that exists in the universe. And here you have a narrow band representing everything we can hear. We can't hear all sounds that exist even on our own planet. So there's a lot of the universe and a lot of our surroundings that we simply can't detect using what we have built onto us to detect sight and sound. So here represents, here the whole graph represents, actually this indicates that there's more before this, more below this. But this graph represents something of the whole spectrum of light and sound. And then this little rectangle, tiny sliver of a rectangle represents everything we are capable of perceiving. So this really limits our access to literal truth. And the interest is I should say, our brains produce a virtual reality for us out of the information it receives from our eyes, from our ears, and all of our sensory organs. Our brain takes all of that information that we can perceive with our sensory organs and creates a virtual reality in our brain for us. And this virtuality is a functional truth. It's a model of the actual universe that is incomplete. It's just good enough to enable us to survive on our home planet. And the interesting thing is other animals live in a completely different functional truth than we do sometimes, because they can perceive different segments of the natural world than we can perceive. Their virtual reality, their subjective experience of what the world is like is different from ours, even if they exist in the exact same place at the exact same time as us. The bee flying around your head on your porch is living in a different version of this world than you are, not just because, it understands life as a bee, but also because what it perceives of the world around you, it has perceptual access to different segments of the reality around it than you have. And one example of this is light. So this is the light spectrum that you can see, and this is the light spectrum that a bee can see. There's parts of the spectrum, the light spectrum, that you can see that are completely invisible to the bee. But there's other parts of the light spectrum that the bee can see that are completely invisible to you. So it is accessing a different segment. It's perceiving a different segment of the world around it than you are. And so its functional truth is going to be different. Its virtual reality will be different. Giraffes, they talk, did you know? They communicate across extraordinarily long distances with loud, booming voices that carry many miles. And you may not have known this because they communicate at a frequency that's outside of what we can hear. Doesn't mean it's quiet. It's not quiet. It's not soft. It's loud and booming, but your ears aren't designed to pick up sound of that frequency. So we can't hear it, but it's perfectly clear to them. So there are sounds around us that we can't perceive that other animals might, and light around us that we can't perceive that other animals might. But the reverse is also true. We have access to certain elements, to certain, our perception gives us access to certain truths around us that other animals may not have as well. All right, technology. We have built technology that can observe things our biological organs can't. And this expands our understanding of the real truth of the universe, right? I mean, how do we even know that there is a light spectrum and a sound spectrum outside of what we can see? How do we know that, or that we can hear? We measure it with machines designed to be able to pick up those sound and light frequencies. Furthermore, we use our scientific knowledge of these things that are outside our biological perception to build things that work. So we really know they're there. For example, radio waves. That's a frequency of light that's invisible to us. All around us can't see it. But we learned about radio waves and we applied that knowledge to build radios, right? Same thing with microwaves. Can't see microwaves. That's a frequency of light. But you probably have a microwave in your kitchen or have at some point, and it works because microwaves are really there. Same thing with X-rays, right? UV light. Sound waves, ultrasonic frequencies, we can use them to clean sensitive materials, to break bones apart inside a human body, to view babies in utero. For sonar, we can build mouse deterrents and dog whistles. Those kinds of mouse deterrents and dog whistles capitalize on that difference in what we can hear and what those animals can hear. So we can build things that they're able to perceive that we can't, so they don't bother us. So we know that this world outside our perception exists, and once we learn of it, we can apply that knowledge to build things that work. We can test our predictions in this way, and that's how we know that these things are really there, that they have an effect on the natural world. Something that doesn't really exist is not going to have. If something exists, you're going to be able to detect it because it'll affect something, right? If it's there, it's going to have an impact on something. We just have to find the way to detect that impact. But despite all of these technological advances, there are still limits to our technology. We have not completed our technological development yet. We are still growing because our technology still limits us. There's still plenty aspects of reality, of truth that we can't know because of technological limitations. Laws of nature. Because of the physical laws built into our universe, there may be things that we can never know, and humans hate that. We hate to think that there might just be a limit. One example, and we don't know for sure where our limits lie, but some things, pretty strong suggestion that we may never know them. So for example, we may never be able to see the edges of our universe because the edges are expanding faster than the speed of light, or at least at the speed of light. And so how can we send something out to detect it and send that information back to ourselves? We can't send out light to bounce off of it, to look at it and bounce it back to our eyes. No, because it will never catch up with the edge of the universe as it expands. Light is the fastest thing in the universe. Nothing can go faster than the speed of light. That's the current understanding anyway. So there's no way for us to send something out to detect and then bring it back. It will never catch up with that edge. And if we can't see the edge of the universe, that might mean we can never know how the universe began. We might not know certain details about how the universe began, perhaps. So it could be that there are always going to be limits on our access to truth, to certain truths. Our age, and this is both personal age and societal age. Personal age, a single individual cannot live long enough to learn everything we know as a society, right? But also in terms of us as a species, we haven't had time on this planet to learn everything yet, even as an entire species collectively. So in fact, we aren't even certain what the actual natural limits will be, or what, even if we have laws of nature limiting our knowledge. We're so far away from reaching that limit yet. So we are just very young. So all of these things limit our access to literal truth, which means that everything we know and everything we're ever going to know is going to be a functional truth, a model of the actual universe, the best model we can come up with, right? So through exploration, though, through using the scientific method, we can continually update and modify our understanding, our model of the universe, of the natural world, and make it ever more accurate. And this will allow us to travel, to spiral ever closer, closer and closer and closer to the literal truth, or the absolute truth over time. At least that's the goal, right? So everything we understand right now is a working model, a functional truth of what is actually true. And perhaps literal truth will always evade us, but the goal is to cycle ever closer to it, to keep learning more and more, keep updating and adjusting until, and just always getting closer to what that literal truth of the universe really is. And by the way, this is an optical illusion, right? It kind of looks like, you stare at it, it kind of looks like it's moving. Optical illusions play on the functional truth that your brain creates, the imperfect rendering that your brain produces of the natural world. This image does not actually move. It's an imperfect rendering between the literal truth of that image and your brain and your perception. All right, so process of science. Back to science not being a set of facts, but rather a method. What is this method? What is the scientific method? Well, it's a series of logical steps that have been designed to remove human bias, because humans, we get so emotionally attached to certain ideas. They become very precious to us. We're very sentimental. But that's not very good in terms of updating and learning sometimes. It's very good for social cohesion, but not very good in terms of updating our knowledge and coming closer to truth, especially when those truths are uncomfortable for us because they contradict what we've learned in the past. So, scientists have known that this is human bias and emotionality is a problem in learning new truths. We've known this for a long time. And so scientific method was made over decades, centuries, to try to eliminate human bias, human emotionality, and learn more purely and more logically. So here's the set of steps that one is supposed to follow when performing a scientific experiment to learn a scientific truth. You make an observation. You observe something in the natural world around you, and you form a hypothesis. And well, when you make an observation, you might have a question like, why does this happen? What is this thing? Okay, so you have an observation and a question that it inspires. And then your hypothesis is a stab at an answer to that question. Well, I think this probably happens because blank. Sometimes your hypothesis might be a stab in the dark. If you're a researcher, you try to make an educated guess, like you use your expertise or you read the expertise of others to help you form an educated guess. Right? So hypothesis is a stab at an answer to your question. You form a prediction. Well, if my hypothesis is correct, then if I do this, I would expect this to happen, right? That's your prediction. If my hypothesis is correct, when I do this, I would expect to see this. Okay? Then you conduct an experiment around your prediction. You do this, and you see, did this actually happen? Were you right or wrong? So when you do the experiment, you're usually doing this a lot of times. You do this 100 times, and then you collect all the results, and that's collecting your data. And then you analyze it. Did it usually happen? Did it happen 50-50? Did it never happen? And then you form your conclusions. You say, well, my hypothesis was supported by my data. My prediction came to fruition, or my hypothesis was rejected by my data. My predictions did not come to fruition. Okay. And then you repeat. If your hypothesis rejected, move on to a new hypothesis and test that. If your hypothesis was supported, you might think, well, what were the flaws in my experiment? Does it happen every time, or was it a fluke? And you just repeat. All right, let's talk about hypotheses. There is an art to producing a good scientific hypothesis. So a good hypothesis should be highly specific, so that when you're testing the hypothesis, you're testing a very specific mechanism. Do not be afraid of getting your hypothesis wrong. Sometimes we learn through a process of elimination. You have to say, well, this wasn't it, and this wasn't it, and this wasn't it. So even if your hypothesis is wrong, you've still learned something. Eventually, you'll get to the right answer. As long as you're learning something, you've had success. It doesn't matter if your initial hypothesis was right or wrong. And the reason a hypothesis should be so specific is to make it testable. You want a hypothesis to be really specific so you can design a really clean, crisp, clear experiment around it that gives you very clear, easy to interpret data. If you get a whole bunch of messy data that could be interpreted this way or that way in light of your hypothesis, well, then it's just a mess and a headache, and nobody's going to believe you anyway, and maybe you were wrong because you don't know how your biases had room to come in. You want crispness, you want clarity, so you need to be specific. So your hypothesis has to be testable. It must be possible to create an experiment around that hypothesis. Also, you want your hypothesis, in light of clarity, speaking towards clarity, you want to identify what the competing hypothesis is. So a null hypothesis is that nothing's going on. It's the boring hypothesis. That's usually the competing hypothesis. Your alternative hypothesis is the one that you're really interested in. I think that if I take this herbal supplement, I'm going to grow taller or something, right? That's your alternative hypothesis, something exciting, right? That's the one you're excited about. To be honest, we would like to take emotionality out of it, but in all honesty, the alternative hypothesis is the one you're really excited about. And the null hypothesis is the boring one. It's like, if I take this herbal supplement, nothing will happen. That's there's nothing to be seen here, nothing's going on here hypothesis. But you really want to clarify. You want to make a very crisp, clear sentence to clarify each of them, because you're going to design an experiment that will fairly pit the two hypotheses against each other. Right. You need truth to have an opportunity to arise, even if it's not the truth you want, right? All right, so falsifiable. This is perhaps the most important aspect of a good scientific hypothesis. You want your hypothesis to be falsifiable. For a hypothesis to be scientific, you have to be able to imagine collecting data that would refute it, that would refute your hypothesis. If you cannot imagine any data that would refute your hypothesis, that would change your mind, you are not working in the realm of science. And this is one of the important differences between religion and science, because religion functions on faith, right? And people who are religious, they really value faith as a virtue. And there are places where faith is important, right? But not it doesn't really work very well in science because to have faith, you have to continue to believe in the face of contradictory evidence. There's no kind of data a person with faith can imagine collecting that would disrupt their faith and change their mind. And that's just not how science works. In science, we practice skepticism. Faith in a preconceived notion makes tests and studies meaningless. Why would you even do an experiment if you know you aren't going to change your mind regardless of what you find? Or if you can't even imagine a kind of data that would prove you wrong, right? So there's no point in doing a scientific experiment then, right? So that's, you have to have a hypothesis that is falsifiable. You have to set up your experiment cleanly so that you are collecting data that is possible to refute your hypothesis. You have to know in advance what data would support the hypothesis and what data would support the opposing, the contradictory hypothesis, and give both kinds of, set up an experiment where either data could come through, whichever one's correct could come through. In fact, in science, we actually tend to construct the entire experiment around searching for the data that supports our competing hypothesis, right? So that whole, the outline of the scientific method, that makes scientific method seem a little bit complicated, but in fact, we use it every day in our everyday lives. A common example would be the reasoning we use to answer the question, why doesn't this flashlight work? 2 reasonable hypotheses I could think of right away. The batteries are dead or the bulb is burned out. Right? And these are falsifiable hypotheses because you can think of an experiment to test it, and you can easily imagine data that would both support each of these hypotheses or refute them. And let's take a closer look. All right, here's your observation. Flashlight doesn't work. And that inspires you a question. Why doesn't this flashlight work? And you could imagine a hypothesis, batteries are dead. That's usually the first one we go to, right? Batteries are dead, that's why it's not working. All right, let's test it. My prediction is that if it's true that the batteries are dead, when I replace the batteries with if I replace the old batteries with new batteries, the flashlight will work again, right? And then you can test this very easily. Take out the old batteries, put in new ones. Result was flashlight still doesn't work. Your hypothesis is rejected. The problem wasn't in the batteries. Move on to hypothesis 2. Bulb must be burned out. Prediction, if I replace the old bulb with a new one, it will fix the problem. Test it. Replace the bulb. Flashlight works. If it didn't work, and you know that this is a falsifiable hypothesis because if it didn't work, that's the data to refute your hypothesis. And we're all attached to the battery hypothesis, right? Because it's really a pain to go get a bulb. We all have batteries in our house, really easy to come by. It's so much easier when it's just the batteries. So we feel really like attached to the hypothesis that it's the batteries. But easy to collect data that contradicts your preferred hypothesis, right? So that's a good, clean experiment. Good, clean, falsifiable hypothesis with a good, clean experiment. Even if even the truth you don't want, it can show itself, right? Maybe it's the wiring, right? Maybe the light didn't work and it's the wiring. And that brings me to a point, always start with the most likely hypothesis, or maybe in some cases, the easiest hypothesis to test. It just saves you time and trouble, right? Usually it's the batteries, so we don't usually go to replacing the bulbs unless we really have to, and we certainly don't go to testing, opening up the flashlight and testing all the wiring before we've tested the more likely hypothesis that the batteries are just dead. So be methodical, be logical in your progression to solving the problem. All right, so a few more points on the process of science. There are no absolutes. Never say my data my data proved my hypothesis. We don't say that in science. You can't publish that in a paper. You can't prove anything because there's always a chance that you just got your results due to a fluke or user error. That chance might be small, but it's still a non-zero chance. Still possible that it was just coincidence that you got the results that you got. So we describe in science what we understand using degrees of certainty. Data supports our hypothesis with 95% probability, 99% probability, blah, blah, blah. This is what statistics is for. We can calculate our level of certainty for findings by quantifying the noise or the messiness in the data and comparing that noise to the strength of the signal in our data and considering our sample size. So if you run a test on 1 million people, you're going to be a lot more confident in your results than if you just tested 3 people, because with only 3 people, your findings could be mere coincidence, but not likely to be coincidence if you tested 1 million, right? So a larger sample size improves your level of confidence. How clean the data came out. Was it really noisy and messy? Is it hard to find the signal? Or is it really crisp and clear? So these are things that are used to quantify with an actual number your your level of confidence, the percent probability that you were wrong. Okay, so no absolutes. You can never prove anything. You can just support it, and we hopefully are believing in the most likely truth, right, rather than the least likely truth. Okay, repeatability, very important. Other researchers have to be able to repeat the experiment and obtain similar results. You have to be able to repeat the experiment and obtain similar results. And related to that, what if you conduct the experiment in a slightly different way? Do your data still support the same conclusions? So repeatability is essential. If you're the only person in the world who can find what you found, maybe there's user error. Maybe you're not really sure how to do the chemistry or whatever it is you're doing. Maybe it was a fluke, or maybe there's some nefarious motivations. Maybe you're trying to deceive somebody. Maybe you're trying to deceive yourself, motivated reasoning. Whatever the case, if you can't repeat the experiment, you can't trust it. So if something is true, it's going to show itself over and over and over again. That just went ahead all on its own. All right, peer review. Researchers critique each other's work and, in the same field. So if you do an experiment, that's just secret to you until you publish it. But you can't publish it unless a panel of your peers, other experts in the same field, agree that your experiment is sound. So you write up your experiment, you send it off to a journal, and a panel of peers will nitpick every minor detail and pull it apart and tell you to run these different statistical analyses and say these statistics aren't useful for what you're doing, and you're imposing your bias here, and your conclusions aren't supported by the data, and on and on and on. And so they really keep the research that gets through to the journals of high quality. So that's very important as well. Humility. And I know, researchers, we don't usually think of scientists as being very humble, right? I mean, they sort of have a reputation for being arrogant. But when it comes to accepting that we were wrong, the scientific method really asks that we practice humility. It demands that we practice humility. And the scientific field, because if you don't stay updated with changing understanding, then you're going to become outdated as a researcher. So you have to keep up to date. Now, of course, when you've believed something your whole life, when you've staked many major decisions upon that belief, when maybe you've devoted your whole life to a particular hypothesis and trying to turn it and support it enough that it becomes a scientific theory, it's difficult to change one's mind. Scientists are humans too, right? And we do have this saying in research that sometimes you have to wait for the next generation of researchers to take over for a new finding to be really accepted. People become stuck in their ways and their thoughts. We all suffer from pride. We all suffer from fear of change. But through the scientific method, we're pushed to leave outdated, outmoded ways of thinking behind in light of new evidence. If you're a nerd like me, you probably know of XKCD Comics. They're comics for science and computer nerds. And this one's called The Difference. I really like this one. So here, there's a still stick figure pulling a lever, and the stick figure gets zapped by lightning. He recovers for a minute, and then a normal person would think, guess I shouldn't do that. I'm not going to pull that lever again. Scientist thinks, wonder if that happens every time. What if it's just a coincidence that he got struck by lightning when he pulled the lever? The only way to really know is to see if the experiment's repeatable, right? It could have just been a total coincidence, and maybe the lever and the lightning weren't connected at all. But of course, a normal person doesn't really want to risk it and is all right with being wrong for the sake of not risking that happening again. Anyway, I really like that distillation of what the scientific method is like. All right, scientific theory. Now, the scientific definition of theory is very different from the colloquial definition of theory. A scientific theory is much broader in scope than a hypothesis, and it's been supported by a large body of evidence. By many different researchers conducting the experiment in many different ways, the conclusions always come out the same. Some examples of theories are cell theory, which is that all living things are made out of cells. We've looked at all living things we can find under a microscope, and they're always made out of cells. So that's cell theory. Maybe there's a small probability that some living thing out there isn't made of cells, especially if we ever find life on a different planet. Maybe we'll find something totally outside of what we perceived, what we understood before. But based on the evidence we have thus far, cell theory is extraordinarily well evidenced. So far, all living things are made of cells. Germ theory. Disease is caused by microbes, by germs. We used to think it was karma, so it actually, we take it for granted. Now we're in the middle of this pandemic. We know it's a virus. We can detect the virus. We can measure its quantity. We can look at how it spreads. We take it for granted that disease is caused by germs, but people didn't used to think so. They thought it was karma, sin, demons, you name it, anything else, because they didn't know that microbes existed. So how else could they explain disease, right? But now we know. We developed a microscope, and powerful microscopes, and this allowed us to test and see new parts of the world that we couldn't see before. So we updated, and now it's very well evidenced that disease is caused by germs. Particulate inheritance theory. You inherit your DNA from your parents. You inherit your genes that create your physical features from your parents. Modern evolutionary synthesis, we'll go into that in another video, and the theory of relativity. Einstein's relativity, space and time are inseparable, and it also describes how gravity is affected by the fabric of space-time. Okay. These are just some examples. There's other theories out there. I just wanted to make a list of some that might be familiar to you. All right, so hypotheses can be tested using controlled studies. Studies can be observational or experimental. An observational study is done in natural settings. A researcher observes what happens naturally, and the limitations on this is that, when you look at a natural system, there's lots of things going on at the same time, and it's sort of difficult to separate influences, like, well, is this the cause of this, or was this other thing the cause of this? So it's difficult to really pinpoint which processes are affecting which other processes when there's so many processes happening at the same time in natural conditions. So you could get around that by doing an experiment where scientists manipulate a situation. They isolate variables and manipulate them to see what happens. If I do this to this, then what happens? Of course, there's limitations there too, because does what happened in a lab really reflect what will happen in a natural system? So usually we really want to use both and collect data in both ways, and to see what is really true. In both cases, we require a control group or a comparison group, which provides us a frame of reference for the observations. And I'll explain control groups in a moment. So for example, scientists conducted a controlled experiment to test the hypothesis that color has evolved as adaptations to protect animals from predation. We're talking about camouflage here. Camouflage protects animals from predation. So can we test this hypothesis? Well, in this case, the researchers used this mouse to test their hypothesis. This is the same species of mouse, but the mice that live on the beach are white, the color of the sand, and the mice that live inland are brown, the color of the grass and the shrubs. So observation, same species of mouse, but those that live in white sands are white, and those that live in the grassy, shrubby inland are brown. And the question would be why? To be more specific, because we really want a good specific question. You have to take a stab in the dark sometimes, right? A good specific question might be, does coat color affect mouse survival? And you can test this. You could think of a few different ways to test this. You might think, so one hypothesis might be, I think that when a mouse's color matches its background, when it's camouflaged, it is more successful at evading predation, and it survives better. And one prediction might be, if I put a mouse in an environment where it is not camouflaged, it will more likely be eaten than when it is camouflaged to its environment or with its environment. All right, so that's a very specific hypothesis and a very specific prediction, very easy to test. And you can imagine data that would refute your hypothesis. You could say, it looks like these camouflaged mice actually are predated upon equally to the non-camouflage mice, right? That would refute your hypothesis. You could test this in a number of ways, and logistics are difficult for tests sometimes. Every experiment is going to have its unique flaws. There's no such thing as a perfect experiment. For example, you could take the brown mice and put them on the beach, and you could take the white mice and put them inland in the brown environment. You could switch them, and then you could measure predation, but there's logistical problems. How are you going to collect the mice? How are you going to know if they're predated upon or not? What if they run really far away and you can't find them again? You could put GPSs on them. You could put them in you could build enclosures for them so they can't travel very far. But what if that enclosure prevents predators from entering? What if GPS trackers are really expensive or difficult to put on the mice? You could take the mice to an artificial environment in the lab, like a zoo environment. But of course, you couldn't be certain your findings there would represent natural conditions. So again, no such thing as a perfect experiment. However you do your experiment, you will then collect your data, and you'll make a conclusion. You're going to conclude whether the data supported, not proved, but supported your experiment or rejected, or supported your hypothesis or rejected your hypothesis. And then you repeat. If it was rejected, hypothesis number 2 now is up for its turn. Or if it's supported, do another experiment that addresses some of the experimental flaws, the design flaws. If you get the same findings, you might be on your way to a theory that camouflage assists with survival by evading predation. All right. So what these researchers did, they conducted their experiment using mouse models. They used fake mice. They assessed predation by looking at bite marks on the models. Models with tooth marks were marked as predated upon. Obviously, there are flaws in this experiment. Remember, you can't make a theory out of one experiment alone. This is one experiment out of many that has tested the role of camouflage in animal survival. So what they did is they had an experimental group. The experimental group, brown mice models, they put on the beach, on the white sands. And white mouse models, they put inland in the brown shrub and grass. So they mismatched the model color to the environment color. That's the experimental group. But you need a control group to compare your results. Because like, okay, let's say 7 beach models were predated upon and 5 inland mouse models were predated upon. Was that a lot? Is that a little? Is that more than you would expect for a camouflage model, or is that less than you would expect for a camouflage model? You need a control group. A control group is a comparison. It's a frame of reference. So what the control group is are mouse models that are camouflaged to their environment. They represent the natural conditions. So white mouse models on the white beach, brown mouse models on the brown grass, and that is the control group. Experimental group is switched, control group is standard. It represents natural conditions. So what this is a controlled experiment. There are variables that are being controlled here. They have a control group to test the hypothesis that color has evolved as an adaptation to protect animals from predation. And you collect the data. Here's the data. So here's the number of attacks on camouflage models and on non-camouflage models. This is the beach and this is inland. So here you have camouflage models that were attacked on the beach and not camouflage models that were attacked on the beach. So white models on the beach and brown models on the beach. Here you have camouflage models that were attacked inland and not camouflage models that were attacked inland. So the brown models inland and the white models inland, okay? And then you can take a ratio. This is the percent of attacks. This is the data analysis part. So this is where you analyze the data. This is the data, and this is analyzing it to see what it means. 71% of attacks on the beach happened to models that were not camouflaged. So the majority of attacks happened to non-camouflage models, brown models on a white beach. Inland, we have about the same. 76% of attacks happened to not camouflage models. So the white models on the brown grass. So in both cases, the majority of attacks happened to models that were not camouflaged. So that data doesn't prove our hypothesis, but it supports it, that the camouflage models were less likely to be attacked than the non-camouflage models. And that's the scientific method.